Today we are releasing our third series in this episode of The Science Behind Coronavirus. Sadly, we've reached a point in the pandemic I've always feared. As you may recall, as we put out these series in March and April, my greatest fear was this virus would go through what I considered then an inevitable evolution in its life cycle. What I mean by that is that the virus would evolve to adapt to man. It's an intelligent machine. It would mutate and evolve and find better and better ways to enter into the human body through its receptor called the spike into the cell called either the lung or the nose of the receptor called ACE2. My fear has now been realized in the sense that these mutations have occurred. So let's begin about trying to understand the science behind the mutations of the coronavirus. This is a pretty complex subject, so the first thing I need to do is explain how spike works and what actually is mutating. As you may recall, in our previous series on the science behind the coronavirus, the virus has this protein on its surface called spike. And as you could see, this green area around the spike is called the RBD, and it's short for receptor binding domain that actually hits your body. So this is the tip of the spear, and this is what is mutating. But sadly, throughout the body of the spike, we are now seeing mutations. Why is this mutation important? Well, it is this piece that's in the body that comes and attacks the rest of the body. And if this is the cell, let's say this is the lung cell, it is this piece of the RBD that enters this receptor called ACE. So the idea of how this works is a virus comes, hits this ACE receptor, takes its RBD, and enters into the cell, and you get infected. So you now have an infection, and this virus then uses the cell to grow and replicate. We've gone into detail with this in the previous series, but it was important for me to emphasize this particular action, because as this was the fear that we've had, that we will create antibodies that would block this action. So that's what is designed as an antibody that will actually try and block this reaction. And that's what we have as monoclonal antibodies. That's in your body as a convalescent serum if you had a previous infection. That is the vaccine that we're trying to generate, what we call anti-spike vaccines, the Moderna vaccine, the Pfizer vaccine, the Johnson & Johnson vaccine, whose goal is to generate these antibodies to block it. My fear is that this virus will mutate itself so that the antibodies can no longer recognize it and it will still be able to get in. And that is what we call biomutation. Well, why is this particular mutation so dangerous that the South Africans have identified, called 501YV2? There is a special mutation at the toe of this RBD. And let me use the next few slides to explain, going a little deeper into the molecular dynamics of this mutation. So if I were to take this spike and reduce it to the green, and now you see the spike at the green, and if I were to take how the spike looks, and you notice the spike, you can imagine it to be a shoe. And the back is the heel. Here's the arch. Let me draw. Here's the heel, that's the arch, and this is the toe. So think of it as the toe, the arch, and the heel. And it turns out in the viruses that have infected us in the first wave, the heel was the site in which it bound very tightly. And that's how it got in. The red ribbon is the receptor in the human body. We were fortunate to participate with Microsoft, and we create what we call a molecular dynamic modeling. Huge amounts of data through our supercomputer that took every element of this RBD and analyzed its motion, its interaction, its what we call molecular dynamic binding and releasing. And what we discovered, this video, is the simulation of thousands of data points right at the toe. And if you play this video, you will see how the toe actually is repelled. So as you could see from this video, the toe completely flops and moves and disconnects, and there's very little contact between the toe of the virus and the receptor of a human cell. And the video on the right is a three-dimensional structure showing how this interaction occurs. Well, so what happens when the virus evolves and wants to increase its infectivity? It turns out it mutates the toe. Next slide. And here is this mutation, which has now been dubbed K484, because it's at that location of the toe where the mutation occurs, so that it now has much greater binding and timing to the ACE receptor. What's the consequence of that? Well, the consequence of that is the heel and the toe now have these binding points. Now we have increased the infectivity tens of times. Well, if it was just more infective, that would be increased transmission, faster speed, and in fact that's exactly, you will hear how dramatically this has spread from the start of Port Elizabeth, ironically my hometown in South Africa where I was born, all the way up and down the coast of South Africa. But we discovered a disturbing fact, and if I could look at the next slide, that, and this is a little complicated slide, but it alters its shape. So on the right-hand side, you see what happens when you have just a single mutant, which is basically the UK mutant, called the 501Y. There's some shape alteration, but when you actually have a triple mutant, if you look at these red dots inside this blue circle, you will see every red dot, every deep red dot represents a locked position between ACE and RBD. So this virus has changed its shape, changed its configuration. It's equivalent of you taking an egg white, throwing it onto a hot stove or oven, and you'll see obviously egg whites, or how protein changes its configuration. Well, what are the implications of that? As soon as it changes its configuration, the antibodies may be less effective. It's like having a lock and key. The best way to think about this is the virus has figured out how to create a master key. So no matter how many locks you have, it'll find another key. That is viral evolution. The concern, which we've never seen as rapidly as this, is how it actually can make multiple keys of multiple mutations simultaneously in the same virus. That is the new variant that we've seen. And you will hear next in my conversations with Professor Tulio de Oliveira how he and his team were able to find directly not only one mutation, but multiple mutations. And the key of multiple mutations, you will hear, is the consequence of that. What happens to multiple mutations as it relates to convalescent plasma, monoclonal antibodies, and perhaps even vaccines that are focusing on actually blocking antibodies against spike. The power of supercomputing has allowed us to model the virus with the RBD receptor, this thing in green, and the ACE receptor, these ribbons in red, to look at the biology of the human space. It's taken literally terabytes of data where proteins and proteins, I call this a dance of proteins, where they interact with each other. And what you see here are the proteins interacting with each other, and what we do this in a rendering model of three-dimensional space. And what you see is the heel, as you can see at the bottom on the right-hand side, the heel hitting and the toe connecting. And these triangles and circles are the mutations of the amino acids, the proteins themselves that allow contact. What we found was in the very first wave, the toe, there was a negative charge, so they would repel each other. The virus has figured out the most clever way to take the toe and now make it a positively charged amino acid. And we theorize, therefore, the positive and the negative now attract each other and they bind. So the viral evolution is a natural thing. It'll find its way, it's an intelligent machine, to be able to change its amino acids. What we've never seen, or rarely seen, and maybe only through the power of genomics because we have the ability to penetrate this kind of insight, you will hear from Professor Tulio de Oliveira that not one amino acid changes, which is normal, or 2, but he's found 8 amino acid changes, all simultaneously, which means this virus has a propensity to mutate and adapt very quickly to find the weak spots so that it could penetrate further. So I am completely of the view that blocking the virus from entry alone will be insufficient to clear the virus. What we would really need is not just blocking, but once it's in, and I think it's inevitable it'll get in, is to kill that cell in which it has entered and prevent its replication. That is what I mean by T cell as opposed to just an antibody. And you may recall we have 3 elements to our human system: the antibody, the B cell, and the T cell. And if we can actually activate all 3, that's our best chance to protect ourselves against this virus that will find and has found immensely fast the ability to penetrate and enter these cells. We have to counter that by, once it's entered, to clear that cell, kill that virus, kill that factory. I have said, and now scientists have actually shown, that antibodies may not be long-lasting. B cells forget, T cells remember, T cells kill. Antibodies may block, but T cells really matter in order to kill and clear this virus. At the end of the day, that's the hope at the end of this tunnel. We will present the findings and conversations with Dr. Tulio de Oliveira, who made this amazing discovery, the disconcerting science he's discovered, but there is hope at the end of this tunnel. My concern has been about these mutations, and when I read about the first mutation rising out of Port Elizabeth, I knew deep in my heart that that township, in which there are very poor, underserved people living in tight quarters, it was very sad for me to hear of this evolution. And what amazing privilege we have had to hear from himself directly, Dr. Professor Tulio de Oliveira, how he came about this discovery. how the science in South Africa continues to flourish despite the difficulties they've been having with droughts and the economic difficulties and COVID. You will hear in this conversation how not only he discovered it, how he's created the collaboration, how it unfortunately is spread throughout, and the important science that he's also shared, not with other scientists, but also with the World Health Organization. What a pleasure it is for us today to welcome a world-leading scientist, a geneticist, and colleague at Sutton, South Africa, Professor Tulio de Oliveira. He's a geneticist that has actually been leading the team in South Africa that's made the groundbreaking discovery of these mutants and these variants, which in fact is now spreading across the world and affecting really our concerns. And it's such a pleasure today to welcome him so that we can actually discuss not only his findings, but what it means to us and what it means to this world. But before we start, obviously, as full disclosure, my company, ImmunityBio, is developing a COVID-19 vaccine. And I'm also pleased to say that we will be actually going into the hurricane of these mutations by coming to South Africa. Dr. Tulio, welcome. What a wonderful pleasure to have you.
Yes, good morning to everyone there in Los Angeles. It's middle of the night here in South Africa, but it's a great pleasure to be with you.
You are now known as the virus hunter, and I think a well-deserved credit and acknowledgement to you and your scientific team in South Africa to making this groundbreaking discovery. Can you tell me a little bit what is that about, the idea that you and your team are now being called the virus hunter?
Okay, thank you, Patrick. Yeah, so our team has been working with viruses for many years, for close to 20 years. Of course, we work a lot with HIV, which is a big problem in South Africa. But in the last 5-10 years, I participate in most of the discovery of a lot of the previous epidemics that affect the world, and many of your viewers will remember the Zika pandemic. We also have a number of yellow fever big outbreaks in Angola and Brazil, and dengue, chikungunya. So one thing that we are learning from the past few years is that emergence of new viruses become quite common, especially as we have degradation of environment and large populations of people in cities and large international travels. Yeah. So what we did, it is that back in January, when we saw the first genome that was released from this virus, the SARS-CoV-2 was released on the 11th of January 2020, we start preparing to set up a very detailed program in South Africa to really follow all the introductions of this virus, but also use that information to try to control outbreaks and to feed back to public health response, which end up us finding a new variant that's much more transmissible in South Africa. But more important, we also help the World Health Organization and international researchers to also find some of the other variants that are circulating in the world. So I think that's why they like to tease us and call us that we are the virus hunters.
Well, I think teasing and actually the seriousness of the work that you do, your group called CRISP, the KwaZulu-Natal group, is really set the stage, frankly, for what even our country should be doing in the United States of pandemic surveillance and pandemic preparedness. Tell us a little bit more about CRISP, because I think credit really must go to not only the South African scientists, but the Medical Research Council and the innovation support that you've gotten from the South African government that has been so forward-looking in building this organization called CRISP and the ability for you to collaborate. So tell us a little bit about your pandemic preparedness and awareness and genomic surveillance that you and I talk about.
Okay, so So around 40 years ago, yeah, I decided to step aside of a big Wellcome Trust Center in South Africa to create our own research institute, which we decided to name CRISP, yeah, which means KwaZulu-Natal Research Innovation and Sequencing Platform. And our objective was really to kind of try to challenge the status quo. And when I say challenge the status quo, we wanted to create a scientific environment that drive the highest level of research in Africa or in South Africa, and in the same way bring innovations to the market, but also reverse the brain drain. Yeah. So one thing that we are very interested in is just to challenge a little bit this whole idea that to do high level science, you have to be associated with international organizations such as the UK or the US, and that you need to be externally funded. And we got major support of our national funders in South Africa, the South African Medical Research Council, and the Technology Innovation Agents to set up a kind of fully automated, basic robotic genomic center, yeah, that would produce and analyze data in real time. And the objective of that is to focus on our own problems, because that was our only main scientific advantage that we had, is that we knew our problems better than the international world. So we decided to use that scientific advantage together with high-level technology to set up a very detailed genomic surveillance for pathogens in South Africa.
Well, I am incredibly proud to be considered still a South African, knowing that your government now, and I'm a little psychotic between American and the South African, so the South African government and the Medical Research Council, and credit to people like Glenda Gray and Slim and Claudina Lotz who have supported. And it's very hard for people to really, in fact, even understand the level and amazing amount of deep science coming out of you and your group and CRISP and the whole concept of pandemic surveillance for the entire Africa. What you do and what the country is now seeing affects the world. And it's such a pleasure to expose the importance of your work, this little team that has actually impacted, maybe actually changed, hopefully, the trajectory of the entire world. So let's go from there. How did this all start? This began in November, where the outbreak started, and you very methodically, with the support of the South African government, has been following this genomic. So could you just describe a little bit historical backwards of how this started, how you found this variant, and how scary you found how quickly it was spreading?
Okay, so we start doing genomic surveillance in South Africa in February 2020. So every week we collected hundreds of samples and we generated the genome, and we knew which variants were circulating in South Africa. We had around 40, 50 variants co-circulating at a given time. And then around the middle of November, we start getting calls from clinicians, medical doctors in this area in South Africa called the Eastern Cape, or the Nelson Mandela Bay, which was a previous city called Port Elizabeth, which I believe that we were born there, Patrick. Yeah. And and the clinicians there, they were very worried because what they have seen, this area has been the heaviest heated area in the country in the first wave. But what they were seeing infections and number of patients arriving in the hospital in a much faster speed than they have seen during this pandemic. So they contact us. Why they contact us? Because we have helped a lot of hospitals in South Africa with outbreak investigations of COVID. I did, and they contact us and they say, like, something strange about this virus. Yeah, it is, people is presenting far too many of them and quite sick. Yeah. And then we decided to take this hint very seriously, and we asked them to start organizing to send us hundreds of samples. At the same time, what we did is to activate our network of labs in the country, which we call the Network for Genomic Surveillance in South Africa, because what we wanted was to sample like close to 50 clinics in that area, sometimes hundreds of kilometers, because we wanted to make sure that what we are seeing in one hospital were representative of the other ones. Yeah. So we got the samples like end of November, and then we prioritized for sequencing. So what we got by 1st of December, we got the first genomes out of the sequencer, and when we analyze, yeah, and I like doing that myself sometimes, we saw this very unusual cluster, very different, yeah, with lots of mutations and very separate from all the other clusters, yeah. And more surprising, every clinic that we sample in that area, it had the same virus, so was not a hospital outbreak, yeah. Then straight away, I went to the chair of our ministerial advisory committee, Professor Salim Abdul Karim. He is our kind of Tony Fauci of South Africa. He has a very similar position as Tony Fauci in the US, and went to him to present and ask permission to him to present also at the World Health Organization Evolution Working Group, which I'm part. And so we did that very early in December. And that's what led also the identification of other major variants in the world, such as the British one. Yeah. So I think from us, we're more like following a very serious collaboration with clinicians and following their advice, and then acting very quick, both on producing and analyzing the genomic data, but then communicating with our government and the international scientific community.
So what you just described is that South Africa, in a funny way, has taken the world's lead in leading the science. It's all the way through the genomic analyses. So you basically have taken absolute advantage of the advancement in science that we understand today, where we can genomically analyze. And I don't know of any other country that has actually taken that lead, looking at the evolution, the life evolution of this virus. As you said, Professor Slim and Glenda Gray and Claudine Lutz, that combination of supporting you and the fact that you are now probably one of the key players in the collaboration. I'm not proud of the fact that it came from Port Elizabeth, but I also understand. I grew up in Port Elizabeth and the townships there in which the people live in very difficult conditions, and some of the immunosuppressed ill patients. So I think I felt so strongly, Tulio, that people recognize the credit when we talk about the 501Y, and we'll get more into the details of these variants. The 501Y variant, so to speak, version 1 in the UK, is in essence being the alarm bells that you sounded, your team sounded, and UK confirmed it, and then you found the version 2, which is a very scary variant, which we'll get into, of the 3 mutations rather than just the 1 mutation. Before we go there, let's talk a little bit about the status of the country, because as you began to sort of see this from a clinical perspective, the spread from the Eastern Cape, people may not know the geography of South Africa, and maybe as we get into the slides, you'll sort of show us a map of the enormity and the spread, not only up and down South Africa, but into Africa and then the rest of the world. Could you talk a little bit about that? How is it now in South Africa, and how is it spread?
Okay, so it's easy to compare what happened in the US. Yeah. So, for example, our first wave in South Africa was very similar to what happened in the United States of America. Yeah, it started in a state like such in the US, in New York, in South Africa was in the Western Cape, in Cape Town, also very touristic town, and start slowly moving from state to state to state. Okay, what in South Africa we call provinces. Yeah. But the second wave in South Africa with this much more transmissible variant, that didn't happen. It is starting one geographical area, and within like a few weeks, just spread everywhere by the coast, really, really, really fast. Yeah, and then enter the center of the country, and now it has spread also to neighboring countries. So the speed of diffusion, that's what you call scientifically, this variant was extremely high, very, very, very fast. Yeah. And we haven't seen that in the first wave. And so what we start, to be honest, we get a little bit scared because like today, I just deposit another 100 genomes or 120 genomes in the public database from KwaZulu-Natal. And from the 120, like 116 were this variant, yeah. And like a month ago, in KwaZulu-Natal, we had like maybe 40% this variant, yeah. So the rate of spread is phenomenal, and the way as it gets to completely dominate, almost like displacing all the previous lineages that exist in the country.
That is very frightening. Because sadly, Tulio, you and I, we talked about this. This is almost inevitable evolution. As we talked about, the virus has now found a path in man. It's used the man as the host. And in order for it to survive, and it's this intelligent machine, it will adapt. It will adapt and adopt and mutate. The speed of this mutation is what I think surprises us, and thank God you discovered it early. But I think the other thing that's really concerning is also the speed of multiple mutations in the same virus. And we'll talk about the implications of that and how it affects convalescent serum and how it affects maybe the antibodies and how it affects into the vaccines. And we'll talk about a little bit about these variants that are growing and fast is all in this receptor binding domain and the spike protein, which is where the virus attacks to enter. So it's what you're saying then is not only the rapid mutations, multiple mutations, but increasing transmissibility and therefore infectivity. We'll talk a little bit about how it affects our treatment. So speak about your concern about the transmissibility and the multiple mutations and the fact that this is at the spike level, and the lay people don't fully understand what spike means. But
Okay, let me explain in simple terms. One thing that concerned the process that we call convergent evolution, what it means that we had these mutations appearing independently in 3 different geographic areas in the world on completely distinct lineages of SARS-CoV-2. So, for example, the virus that we found in South Africa has this very important mutation, the 501Y, that increased transmissibility, which this mutation also emerged in another independent lineage in London, which that lineage, what they call the B117, it now have been found in 50 different countries because London is much more connected internationally than South Africa. Yeah. The same mutation, the 501Y, was found in travelers from the Amazon that came from the Amazon in Brazil to Japan. Yeah. And so again, completely independent. And then what got even more scary is that not only this 501 mutation, they also sometimes have what you call a nasty brother, yeah, that's very close by, which is another mutation that's called the 484K. And these mutations seem to really be escape of antibodies. Yeah, what it means that maybe the body defense will be quite weaker against this virus. And that mutation emerged in the virus in South Africa in combination with the 501Y and also the virus in Brazil. And we are not going to be surprised in the next 2 weeks, we find like 10 of these variants, because if that can emerge in South America, in Europe, and in Africa, yeah, why will not emerge in other places that haven't controlled the pandemic, such as United States of America?
So I can explain this to the public. What you're actually saying is even scarier, that this was not a transmission possibly from South Africa. This is what you call convergent evolution, meaning that the virus itself, as an intelligent machine, adapts. And now you talk about the bad brother or the nasty brother. So, and we will talk about it, you and I have been talking about it in great detail, and we shared with you our molecular dynamic analyses. where if I would describe to lay terms this virus that RBD or spike protein that enters into the ACE, is you consider it a shoe or a high-heeled shoe with a heel, an arch, and a toe. And the 501 is the mutation at the heel. But that heel was always tight to the ACE2 receptor. So that was the first variant that the UK, and in fact you alerted that. Then you discovered the toe, which is the 484. Now when you have the heel and toe combined, that changes the shape. You now have a shape changer, and that is what is now scary, because those, you think, mutations not only is convergently developing, will develop across the world, but and we'll get to that in more detail as we discuss this together, what it does with regard to convalescent serum and vaccines and even the current treatment we have. So this intelligent machine is remarkable, and I've been saying, Tulio, your variant, the 501 versus V1 and the 501 V2, should really be named now 484 V1, because that is what I believe you as a group have really changed the awareness of this world. Maybe we can talk a little bit more of that scientifically, because I think people will understand what you call this triple mutation and how does this variant work, what does it do, and what does it do to the treatments that we have. What does it mean that you have a shapeshifter, a conformational change in scientific lingo, or the change of the lock and key in the language of a layperson? Could you just talk a little bit about that?
So what we found is that these mutations, as you described, the heel, the arch, and the toe, they make this lock much stronger. Not only they make much stronger, but the virus can enter easier to the body, but then they will potentially, and we have find evidence, they will block the antibodies that would stop that to happen. Yeah. So we may be ending up with viruses around the world that are much better adapted for transmission between humans and to evade the immunological machine of the body, that's kind of the fighters or the soldiers of the body, to affect that. Of course, this is worry, because one of the things that the vaccines try to do is to mimic or stimulate the defense of the body.
Tulio, thank you for describing that thing about multidrug resistance and the mutations that you're working all your life there in Africa and South Africa. From my perspective, this is not unique biology in the sense that in cancer, we see these mutations happening and they become, you actually generate a mutation in response to the treatment to become resistant to that treatment, and that's how cancer evolves, so that you ultimately get to sadly, no treatment exists unless your immune system kills it, i.e. the T cell, and we'll get to that. When you get to multidrug resistance, whether it be HIV, malaria, TB, that's the same thing happens. These biological systems have evolved almost inevitably to mutate, to adapt to the treatment that we as human beings induce. So it appears that this coronavirus, which is already deadly, has done exactly that, is mutated incredibly, incredibly fast. Will you now just address a little bit, and I know this is very scary information, but it's important coming from the scientists that have done the work, yourself and Alex and Richard. And we've had the privilege of actually seeing your science before it's even published, and I assume that the paper is about to be published in Nature or somewhere soon. Could you describe and talk a little bit about how this variant, what we call now, there's multiple variants, but we'll just call it the 484 variant, your variant, the V2 variant, same number, with this multiple mutation of the toe, how it compares when you tested the convalescent serum against that. Will you describe how it grows and how it grows differently? You already talked about how it spreads. So if you could just describe your findings that are basically being published this week or next week.
Okay, so of course that is not just our findings, yeah, and it's what we called like that all the evidence is pointing the same direction, yeah. So once we highlight these mutations, the 501Y and the 484, yeah, hundreds of scientific groups around the world start doing experiments for test these mutations, including also your own company, Patrick, also modeling that in the molecular dynamics. Yeah. And one thing that's becoming really evident from many lines of evidence, what it means from protein structure, how the virus bind and antibodies cannot get into the space, from what you call pseudovirus. They have a fantastic work that was published last week from Jesse Bloom lab that's in the Fred Hutchinson in Seattle, showing that this mutation, the 484, will be the most important mutation to escape neutralization. They also had another fantastic group in Italy that keep exposing the virus to convalescent plasma. And around 80 days later, this exposure, it developed the 484 mutation and escaped the neutralization. But what was interesting, it is what we call like reproducible science or high-level science, when many different groups, independent in the world, using different technology, can come to a very similar conclusion. And then the main take-home message is that if we leave this coronavirus circulating for so long at such a high level, that this virus will outsmart us.
It's a very, very, very disconcerting because it's basically man against virus. It's virus against man, almost. Not I'm going to the level of existential, because we're going to find a way to win this war. So let me just interpret what you just said again. What is really important and exciting is that really completely independent, high-level, credible scientists have not only corroborated, but actually independently discovered the same path. On the one hand, the Fred Hutch group showed using the pseudovirus that the E484 actually is important. I think the most scary and concerning one is a group also in Italy that showed that when you put viruses in convalescent plasma, they themselves independently evolve to super select the 484, which becomes what you call this escape mutant. And then finally, all of this work you had already earlier confirmed in December when we did this combination. And when you put these combinations together, as you said, not only does it lock tightly and in effect higher, it shapeshifts, it conformally changes. Could you describe a little bit the experiments you did there? The growth, the syncytial growth of those cells. And then we'll talk about the slides, which I saw, which was very frightening, of the virus not being affected by first wave plasma.
Okay, so I can describe the part of the experiment. So what we did quite early, the basic a day after we discovered this variant, that we discussed with the chair of our ethics board, about linking to another group in the same building as us from Professor Alex Siegel, and then we submit and we got permission from our ethics board to give them virus isolates or samples from individuals so they could grow that in the lab, and it was quite phenomenal because they got the virus like basic 10 or 15 of December, and that when the holiday season started in South Africa. But what this group did, that they just didn't took any holiday. They just, on Christmas Day, on New Year Day, they were there in the lab, because this is very advanced labs. It's what you call a P3 lab. It's the highest security bio lab that you have to go, like even with respirators, you look like as a kind that you are in the space, with a suit, So what they do, they have these different cells, lung cells and lab cells called Vero6 that they go and they infect them with the virus. And normally take around 2 weeks or to the virus start have enough titer to grow. After the virus grow in these cells, what they do, they put a kind of membrane on top, so the virus cannot they cannot spread to the other side. So what they do after that, they can count. They use these very advanced microscopes, and they can count the number of particles of the virus and what they call the colony forming units. Yeah. And so because that they know how many virus will circulate, will grow without any antibody. And then what they do, they start titrating the antibodies of the convalescent plasma. And they do that in a combination like a dilution of 1 to 100, 1 to 200, 1 to 400, 1 to 800. It's a very methodical process that you stay exposing the virus that's grow to different concentrations of the plasma or the covalent serum, yeah, and see and then counting how many virus get neutralized, yeah. But what you have to do that not only with the virus that of this new variant, but we also have to do that with the virus that was previous circulating, yeah. And then you measure the difference between the two. And what the difference came is quite striking, is that the previous variants that were circulating could be well neutralized, and the new one was very weakly neutralized. Yeah, and some individuals would treat, or the plasma of the individual would treat this virus as naive person, as someone that had never seen the virus. Yeah, what's quite worrying because that has major repercussions for potential reinfection, but also to some of the component of the response of vaccines.
Well, what you just brought up here, again, another level of seminal work. One, at first, it shows the collaboration, the importance of high-level scientific collaboration in the same building. You and I talked about that in this, and again, I'm so proud of the country and yourselves. But the profound implications that your work now implies and have demonstrated is that patients who were previously infected, their convalescent serum may not protect them from this next wave of this virus. That's number one. Number two, the potential for them getting reinfected is therefore potential. And number three, the monoclonal antibodies that are being developed or are in use, and EUA may not be effective, the Regeneron and Eli Lilly against this virus. And number four, we don't know yet whether the vaccines that are focusing on spike may be infected. So there's a huge amount of implications up and downstream. So without creating this fear, we obviously need to learn this. But I've also understood that you've discovered a patient that was infected with the first wave and now reinfected with the second virus. Could you talk a little bit about that patient, if I may?
Okay, so first, that's what you highlight there. That's the excitement of science. Each question that you answer, it normally comes 4 or 5 different questions to answer after that. Yeah. So all these questions are extremely important. And again, we are working with a big consortium of national and international scientists to try to answer them very quick. Yeah. Yes, we do have a number of patients. I think that we have 5. By today, we confirmed 5 different patients that were infected with a previous virus that was around like 5-6 months ago, and they became reinfected with this new variant. Yeah, and that of course it is worrying. Yeah, we are still going quite methodically through the data, because one thing that we still need to know is that, okay, these 5 patients, that a rare event or is a common event. Yeah. So one thing that we know is that they can get reinfected with this virus, but we have to understand what is how common this is, because so far everyone assumed that reinfection was extremely uncommon. Yeah. And anyway, to prove reinfection is quite challenging. You need to have a very good, well-functioning genomic surveillance program, because you have to have a genome of the virus that infected the person 6 months, and another one 6 months later. So you have to really be good to generate data and have good sample control. And we also have to do the human marker, make sure that the samples are from the same individual. Yeah. So that's the first question. How common is reinfection? We know that this virus can reinfect people, but the next question is how common? The other next important question is that, is the reinfection with less severe clinical symptoms or similar clinical symptoms? So at the moment we are working on that, and so far it seems that maybe the reinfection may be quite similar or slightly less clinical symptoms, but it's early day. We have to go through all the clinical file. But one thing that we noticed that got us quite worried, was also the psychological effect, because these individuals, they are mostly healthcare workers, and one of them had to be hospitalized from most, like, not only from the symptoms, but really nervous about being reinfected and bringing this virus back to the family, because they assumed that now was naturally protected. And that's something that we worry a lot. Our healthcare workers, not only in South Africa, but around the world, they are really stretched to the limit. And they don't need like some more fear of reinfection with a variant that may have evolved to make that more often.
You know, what you raise again every time is exactly what when you do good science, one question gets another question or many other questions. But what you just said in your last statement is a little, very actually even more disconcerting. So this patient was a healthcare worker, as I understand it. She got infected with the first wave, recovered, completely negative, went back to work, got reinfected, but now with the second wave. You don't know whether she had a vaccine or anything in between, but what you're saying is that she got reinfected by the second wave, meaning that her whatever antibodies that she had from the first wave didn't protect her. Is that what the implication of that, even though it was just one patient now at this point?
Yes. But that we also know from the previous variants that some individual will have, after many months, the antibody levels will decrease, and then there is potential of reinfection. One thing that I think that especially healthcare workers, they get very, very nervous. Is not only about themselves, but also to bring the infection back home. And infecting the families. So I think that's one thing that add a lot to the anxiety of healthcare workers. Not they are in this terrible situation that every day they are seeing people like desperate need the oxygen and dying in the ICU wards. But they also worry about themselves and about the families. So if some healthcare workers, if they have been, and it's quite common that this virus in certain hospitals, not only in South Africa, but across the world, would have infected 20, 30, 40% of the healthcare workers. They're extremely transmissible in a hospital environment. But then if you have passed through that, and you got sick, and then, okay, you've thought that now you're immune, and then you get in that situation again, it's very disconcerting, and it's a very nervous situation to be. So that's why we think that and we really hope that these reinfections are a rare case. We have our fingers crossed that they are not as normal as we expect, because we do worry about the frontline healthcare workers.
So Tulio, one of the premises of the Second Opinion at LA Times is to provide hope. What we've been talking about is really, really, really scary. So in your opinion, as we move forward here, knowing that potentially that convalescent antibodies don't work, protective antibodies aren't protective, monoclonal antibodies may not be currently protective, vaccines that just focus on spike may not be protective as an antibody. What do you think is the solution here? Where do you think we're going with this? What is the hope? What's the hope for the future? And we need to move fast for not only for ourselves, but for the world, frankly, not only just in South Africa, where it's now rampant. What do you think is the hope we can provide to the world of where we should go now that we know that the antibodies, the shape-shifting happens?
Okay. So I am an extremely positive person, and I have a lot of hope. Yeah. So one thing that we can see it's what we hope that increases a little bit of shifting of the globe and certain countries, and we hope also United States of America taking more serious this virus. Yeah. Because one thing is that it's our utmost importance to try to these three variants that we describe here to be a basic wake-up call to the world and government and public health officials to take very serious and try to decrease transmission to almost zero. Yeah, it's also a wake-up call to very quick increase the number and the breadth of vaccines that have been developed and distribute them as quick as possible. Because one thing that we know is that even that some of these variants may be less neutralized by antibodies, the vaccines a lot of times they have broadly neutralized antibodies, and they will act in many different areas of the virus, and then also have the other part of the immune system as the T cells that could also hit on this infection. Yeah. So also increase the delivery of vaccines and bring more potent vaccines, try to decrease transmission, and also realize the world that we are in a global pandemic and we need a global response, because just controlling infections in your own country or not, it means that this virus spread very fast. For example, the variant that was found in the UK, the B117, now is in 50 different countries. The South African one, by this evening in South Africa, when you got a look on the data, was in 16 different countries. Yeah, and so these lineages, they spread very fast. So the world has to try to focus on controlling transmission everywhere, and also do that not only in the rich countries but also in the developing and in countries. So to try to protect the whole community. So I really hope that's a wake up call, and that we can really now use the weapons that we have to control this pandemic, which we have many of them. We just have to use them more effectively and more focus.
Let's close with this one concern about the cold chain and Africa. And that really is my personal concern, right? Developing countries require, again, to talk about thinking out of the box, is to develop an oral vaccine or something that we did for polio, where we had a little sugar tablet under the tongue. What's your thoughts about that? Because this minus 80 degrees mRNA, I get that, it's new technology. But I don't see that being a practical approach for poor countries in Africa.
Yes. It's absolutely not a practical approach. For example, in my lab, we do have many minus 80 freezers, but that's extremely unusual. Yeah, and to have that in South Africa or in Africa. I must also say that in South Africa at the moment, we have a massive problem with our energy supply. So every day we run out of electricity for 2 to 4 hours a day. So even any kind of refrigeration, especially in rural setting, may be challenging. Yeah. And I would agree with you that a vaccine should be effective in developing countries, but also in developed countries, because it would be much easier to administrate, would be fantastic if it do not need cold chain and also do not need injection. Yeah, because it would be much much better that could be taken as a pill, or as you say, as the polio, which is a little drop in the mouth. So, of course, in South Africa is by far the most developed country in Africa. Yeah. And South Africa, many of your viewers may not know, but it's even part of the G20 nations, the 20 most developed countries in the world. Yeah. And we wouldn't have the facilities to distribute a vaccine that needs minus 80 or even minus 30. That's the second level of ultra deep freezer. Yeah. And this would be much less available in the other countries in Africa, but also in Southeast Asia and also in Latin America. So I do agree with you that the technology have not only to develop vaccines, but also have to use technology to make them easily to distribute.
Well, Tulio, I can't tell you how grateful I am for you to present to the Los Angeles Times your insight. I'm also can't tell you how grateful I am to you and your team for really making not only groundbreaking, world-shattering, enlightening science that's going to affect the world. And I'm also can't tell you how proud I am of the South African research community and the government that supported you. And thank you for being with us today. As I said from the beginning, your work and your scientific team and the science that South Africa has performed is hallmark and will have major impact on our fight against COVID. So thank you for your time. I really appreciate this conversation. We've discussed the concept of mutations. We've shown how this triple mutant binds. I now need to get to the point of this section of discussion of what does it mean. The concern to all of us is, if you have this mutation, does it mean that antibodies are no longer effective? Does it mean that convalescent serum is no longer effective? Does it mean that you could get in reinfection? And even of greatest concern, does it mean that the vaccines won't prevent us from getting infected by this mutation? I'm pleased to say that Dr. Tulio de Oliveira today has broken some news on this subject, and I will ask him to present this subject matter in a lecture presentation to us so we can hear directly from him his findings on the ineffectiveness of convalescent serum. Tulio, please go ahead.
Just some simple information. One of the main reasons why in South Africa, both myself and my colleagues, we look very careful at mutations is because for the past 20 years, we have been looking at mutations on HIV and TB, tuberculosis, that normally associated with a resistance to treatment, what we call multidrug resistance pathogens. So we got well trained on infectious diseases of looking for mutations and try to understand how they work. Yeah, we have been working basic day and night in South Africa and many colleagues around the world during all the holiday season to try to understand more how these mutations interact, but also how the body cell defense would be effective or not against this virus. So this presentation is just highlighting a little bit the emergence and rapid spread of a new SARS-CoV-2 lineage or variant with multiple spike mutations in South Africa. Yeah, and then just to start, it's always good to highlight the tool that we are using. One of the main tools is called genomics. What it means is to generate genomes of the virus. Yeah, and that has been such a potent tool in COVID-19 fight. It was used to discover the virus that caused the disease, yeah, which was the SARS-CoV-2. It was, and we did that, and the scientific community did that in weeks, yeah. And then it was used to develop diagnostics, yeah, was also used to develop vaccines, was used to track transmission, to identify infection, to understand virus and host interaction, and lately to identify new variants that transmit very fast. So genomics has been one of the cornerstones of this response to the pandemic, which over 350,000 genomes produced, with 3.5 of those in South Africa. Yeah. So today, as I speak to you, just came. We have been featured in the main scientific magazine, one of the main ones, Nature. Yeah, about how labs, and especially our labs, have been really scrambled or being like working really hard to identify these variants and to also understand how they arise and what's the effect. And that's it. It is a picture of a patient in a South African hospital. So what we did in South Africa with initial funding from the Department of Science and Innovation, the South African Medical Research Council, is to set up a network of genomic surveillance. Yeah. And how we did that is to set up like 5 labs that across the country to sequence the virus in a very comprehensive way in South Africa. So CRISP is the organization that I led. It's leading this network. And another thing that we did is to put this genomics lab always next to a national health laboratory service. So that's the labs that serve all the health facilities. So data and samples could go very quick from diagnostic to sequencing and then feedback to clinicians. Yeah. And that's what we found. We found this is a paper that's also coming this week. That's from the first wave of infection in South Africa. Yeah. And then just to highlight here, we had quite a severe first wave, not close how severe is the second wave. But what we did is to start genotyping the virus very early. We even got the contact of the first infection in the country. And one thing that we learned is where the virus comes from. Most of the virus came from introduced in Africa through Europe, some of them from North America, but the great majority from Europe. Why Europe? 80% of the international air traffic outside the continent in South Africa is through Europe. And then these introductions now showing black set up local transmission. So despite we had hundreds of introductions from outside, we end up with more than 99% of the infections in South Africa being transmitted within the country and by citizens to each other. Yeah. And that's what we found. Yeah. Again, we start that very early in February. That's the data. And here's the proportion of the genomes. When I have white, it's not that I don't have data, it's the opposite. Normally I have 20, 30 lineages there. So just for simplicity, I'm showing in white. So what we had normally, South Africa have 20, 30, 40 lineages circulating. Around May, we start creating what you call our own lineages, yeah, which they get a number in the classification, and they are mostly created through super spreading events such as hospital outbreaks or convention center outbreak. They follow in the population. And these lineages, they were circulating with a small prevalence, 10, 20%. Yeah? But then what we found after a tip from some doctors in the Nelson Mandela Bay, it is a new lineage or new variant, which we number 501YV2. And this variant appear, we first samples that we get was in the middle of November. We went back to the freezers to see when it start appearing. And as it appears, start completely dominating and displacing all the other lineages, yeah? And if I show data that came today from our lab, now is in 99% of our virus. Is only one virus circulating in the country. And that is this lineage. And that's one of the main data that could show us that this variant is much more transmissible. Not only it completely dominates, but it is traveling very, very, very fast into many different areas. Yeah. So here's just a map.
Tulio, do you mind going back to that last slide? Because I think that slide is so important that you produce for us to explain to the lay audience. So the yellow bar that started in October, if you look by September, there basically wasn't any of that yellow bar, which is the variant that you now discovered, called, I call that your 484 variant. And this is all just South Africa. So by what you're saying, by November, that yellow bar had taken over the proportion of genomes of infections, 90% that the yellow bar speaks to the rapidity of the infection transmissibility and what I call genomic surveillance. Really credit to your strength of genomic surveillance and the importance of pandemic surveillance and pandemic awareness. Very much like battlefield awareness. You need to know what enemy you're fighting.
Yes, thank you, Patrick. And of course, that also the way that we did is to do something that sometimes people think that's boring, but it is repetitive. Every week, get hundreds of samples from dozens and dozens of clinics and do the analysis. Yeah, and that sometimes is what's not the exciting part on science. But if you do that very repetitive and consistent, you can see change very quick. Yeah. So if I just go to the next slide, because I also find that one quite exciting. Yeah. What we can do here, and that's these lines. Yeah. And you're gonna see the different shades. The shades we can recreate the travel of this virus. Yeah. So we can identify where it started in the Nelson Mandela Bay, which was called Port Elizabeth. Yeah, around like not only was first detected in September, but all these multiple introductions to next towns. Yeah, and then which one of them quite early in blue, it go to this area in South Africa, very touristic area, the Garden Route, fantastic, beautiful place. And that it also serves as an amplification event because lots of people travel to this area to introduce the virus much further away. Yeah? And South Africa is quite a big country. To drive from KwaZulu-Natal to Cape Town is like 2,500 kilometers, quite far. Not as big as the US or what would be like 1.7 thousand miles. Yeah, but it's quite far, and the speed of the diffusion of this virus I'm going to show you in the next slide. Yeah. Also, the power of do that kind of analysis thoroughly. Yeah. And what we did is to sample over 100 clinics. Yeah. So just to make sure that what we are seeing is being replicated, and that's the kind of suggestion that I have for United States of America to also create a network to do weekly surveillance, but also across the different states. Yeah. And this is the graphic that shocked us a little bit. Yeah. Here is the Nelson Mandela Bay. And the different shades in red is what you call test positivity. Yeah? And the test positivity in that area around October was getting closer to 20%. What it means, every 5 tests, one is positive. The World Health Organization considered a pandemic when the test positivity passed 5%. Let's just see the speed of diffusion of this virus across the coast. Yeah. So here, just moving through the weeks, and you're gonna see many of the areas become very dark red. Yeah? And this it means that all these areas, every 10 tests, more than 3 are positive. Yeah? What it means is that the size of the pandemic is multiple times higher. And we have never seen that. The speed in 10 weeks moving to all the coast and completely dominating all the areas with very high level of infections. And let's just look at that again. Yeah. You can see moving from the center, going to the south, east, west, and the northeast very, very quick. Yeah. And we didn't see that in the first wave. Yeah. If you go to the next slide, and here again, put in the whole country, you can see that by January, not only the same thing happened in the center of the country, but a lot in the districts that border the neighboring countries. Here would be Swaziland, Mozambique, Zimbabwe, Botswana, Namibia, Lesotho. So we have every reason to believe that these variants now the main dominant variants in all the neighboring countries. Botswana, the last genomes that they did, 100% of them were this variant. We just received 150 samples from Mozambique, and next week we will know exactly what's the frequency of that variant. But given that all the neighboring districts have this very high unusual test positivity, we are expecting most of them to be this variant, especially because during the Christmas period, a lot of migrant workers from the country, South Africa is the powerhouse economically, would go back to the countries to spend the time with the families. Yeah. So how different is this variant? Yeah, this variant, as Patrick mentioned, that's compared with the previous three variants that were circulating at around 10% each in South Africa, or 15%. It has double of mutations, like 23. The other ones had like 10, 11, 12. Yeah. But more worry that not only has double of the mutation, but most of the mutations, they cause amino acid changes. And that's when it changes the function of the protein. Yeah. And when we look at where that's based, yeah, as the previous lineages, they had 2 or 3 mutations in the spike protein. That's the most important protein of the virus that will bind the receptor to enter. This one has either 7 or 8 mutations. And all these mutations will cause amino acid mutations, yeah, where all the other ones only have one mutation, the spike gene, the 614G, which has been shown to increase transmissibility, but this one has either 7 or 8. Yeah. And then more important is to look at this mutation, the 3-dimension structure. That's something that which we show the protein. So here in light green, we have what you call the receptor binding domain, which the mutation that's shown to increase transmissibility, the 501Y. And here is the what we call the very bad brother, the 484K, because these mutations here in the receptor binding domain is the main area that's affected by antibodies. Yeah. This is neutralized antibodies, which is elicited during SARS-CoV-2 infection, and it's 90% of the neutralizing activities present in convalescent sera. Yeah. But we also see another cluster of mutations here that maybe we wouldn't see then if you look at linearly, such as 18, 8215, and a deletion. But this is also in another important area of the virus, the N-terminal domain. That's the other area that's affected by antibodies. Yeah. So this virus seems to have like multiple mutations and a deletion in the spike protein, exactly where you don't want it to have in the main domains for both receptor, but also the N-terminal domain. Yeah. So that's where we get worried when all the different groups in the world start studying these mutations. Yeah. In this graphic here, I have it sound complicated, but it is not. If it's dark red, it means that there is resistance to the different antibodies, the antibody names on the top, and here is the position of the mutations. Yeah. So you're going to see this big cluster of red around the 484, especially the 484K, because this is the mutation that showed to confer the broader cross-resistance to several monoclonal antibodies and even combinations of them. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And here we see 6 of them with completely completely resistance, and the remaining ones with kind of high level resistance above 50%. Yeah. And that's the number of different papers that highlight that. Another crucial experiment comes from the group in Italy that they grow this virus in the lab, and they keep putting convalescent plasma, and around 11 weeks or day 80, it developed the E84K, and that's a massive decrease in neutralization. It's followed by insertion and basic. This now it's a completely immune-escape mutation. So this E484K led to a 4-fold decrease in neutralizing activity, and this with 19 different convalescent plasma. So the virus in the bottom, here just to notice that's exactly the opposite. Light white, it's low neutralizing activity. And as you see, most of the antibodies do not seem to be working. That was the crucial results that came last week, also from Bloom Lab, from the Fred Hunch, yeah, which what he do, he map all the mutations in the receptor binding domain, so they are here. And what it showed that the mutation that have the highest escape across the cell, this means that the one that reduced the bind activity is the 484, yeah. So it caused the largest drop in neutralization. It's easy to understand because this is a position that highly exposed there. So once it changes, it gets less affected by antibodies. So again, all this evidence is just building in the last week, yeah, of this mutation being very important. But we want to highlight that today was another preprint that came about the N-terminal domain, showing that a lot of the mutations that they have in the 501v2 and also in the V3, the one that's found in Brazil, also have the 18F, the 484K, and the 501Y. They seem to also be associated with escape mutations. So this true, and we have a deletion just before here. Also with monoclonal antibodies. I know that everyone is just looking at the receptor binding domain, which probably are very important, the most important region, but would like to highlight that there is also this other region that have cluster of mutations that also support the escape from antibodies. Yeah.
So what you were showing there, if I may, on that, because this is so important for even the scientific community, the regulatory authorities, the NIH, to understand that when you get this thing called the NTD, then the N-terminal domain of the spike protein, it's not the direct interface between the RBD and the ACE2. Yet, when you mutate here, you still would think you will see emergence then of escape from the monoclonal antibodies, meaning that these mutations outside of these neutralizing antibody assays are as important in terms of getting resistance. Is that what you're saying here, Tulio?
We wouldn't know if it is as important, but what we think that they are also important, yeah. And just to highlight, should not forget about them because this NTD area it's also seem to have strong signs to be escape mutants from antibodies, yeah.
Which speaks to the fact that one needs to then find even monoclonal antibodies to the N-terminal domain to see if that would help.
Yes.
Okay, thank you.
So this is the kind of viral cells and the lung cells that are there happily circulating there and being replicating in the lab. Yeah. So see what's happened when these are infected with SARS-CoV-2. When they are infected with SARS-CoV-2, they are circulating, the virus starts infecting them. They start doing what Patrick called very similar to cancer. They start the cell-to-cell spread, and then they start killing the cells very quick. And that's exactly what's happened in the lungs of people. That once the virus circulates there for some time, especially first as cell-free virus, but then when start infecting many cells, they start transmitting to each other and start having massive number of cell deaths, that big organelles that are being created. So once this virus is grown in the lab and you can infect them with the virus, then what you can do, you can use the convalescent plasma from previous infections and see how that will neutralize. And that's what we did here in South Africa. I'm just trying to ground you. I know that many people find it difficult to understand phylogenetic trees, but here's what you call a tip-dated tree. That's the dates where the virus were isolated. So the first thing to see is the big cluster, the 501v2. That's basic, and that's all the previous virus circulating in South Africa. And when this one appear, almost the other ones cease to exist in very small numbers. Yeah. And here, what I'm marking here in orange is the virus that we give to Alex Siegel, which he grew in the lab. And this virus has all these mutations in the receptor binding domain. And here, with also another virus that he has collected from the first wave back in July, yeah, which is also growing in the lab. So he take these three different viruses growing in the lab, and then he take the convalescent plasma from this one, that one, that one, that one, that one, that one, and each other. And then he exposed to each other to see how they neutralize. So that's how the experiments work. Yeah, basic. All these individuals that you have the virus, you also have like blood from them. You know how they respond to the first infection, and you see how the plasma will neutralize this virus. Yeah? And I'm not going to show you all the results. I'm just going to give you a peep on that. So that's how it looks. There you have a focal point. Here is an uninfected cell, so you can see no infection. Here's the virus from the first wave. That's when you just leave to circulate. It's creating all these foci of infection. And then we start titrating and add different convalescent plasma. And here is at low concentration. And then we start increasing the concentration of that. And then what you can see, that start killing most of the virus infection. And you have very little that's alive. But that's what's happened with the 501v2 with all these mutations. Here is without any convalescent plasma. And as you keep adding more convalescent plasma, you do see some neutralization, but much, much less than you expect. Yeah. And that's what we're going to be publishing next week, showing very limited, very little neutralization from convalescent plasma on this new virus, which has all the characteristic of being a escape antibody mutant virus, which is much less neutralized. But again, as I mentioned in the previous slides, this building another 5 or 6 big line of evidence suggesting always that these different mutations, especially the 484K, may allow the virus escape from antibody. Yeah. So just to conclude, yeah, genomic surveillance is a critical component of the public health response and should be scaled in most of the countries in the world. And we think that we can exemplify very well by the early detection and tracking of this new 501YV2 variant. Yeah. We have detected a new lineage with mutation at key sites in spike glycoprotein. This mutation associated with decreased antibody response in many different experiments from our groups and many international groups in the world. From the protein structure, from the pseudovirus, from live virus, all the evidence in point of the same direction of decreased antibody response. We crucially and very quick need to determine the clinical significance, such as the risk of reinfection and the impact of vaccines. Yeah. Just to finish, a lot of the international media will say that I'm the virus hunter and that I have done that alone. But in reality, this is the work of dozens of, if not hundreds of scientists in a very strong, coordinated, collaborative network for genomic surveillance involving all of the top universities in the country and research institutes and funded by the national foundations, both the Department of Science and Innovation and the South Africa Medical Research Council. Yeah. Thank you. And it's a great pleasure to talk to the LA Times and to you, Patrick. Thank you.
Thank you for your time. I really appreciate this conversation, and I look forward to having much more conversations like this. I am sure you'll be sought after, but I really thank you for your time to give us at the LA Times this opportunity.
Yeah, thank you. It's a pleasure. And the serious media has a very important role on this pandemic and to bring science to the forefront of all the public. So thank you, Patrick, and thank you for your editorial team.
Well, I look forward to coming back home to work on this project with you. So keep well, keep safe, and keep working. The scientists of the world are entering into an amazing phase of incredible collaboration and collegiality. We have tools in our hands we've never had before. The genomics analysis that Tulio and his team have done, the capabilities of actually now building next generation vaccines as rapidly as been done with the mRNA vaccines, and the idea now of our insider understanding that what we call immune pressure and viral evolution is here, but we do have tools to fight that. And the tools to fight that is the T cells. You and I have T cells. We have NK cells. We have macrophages. So your immune system is available to fight that. We will not give up. The opportunity for this virus to take over this planet is not at hand. The human race coming together has a way of overcoming, and we will overcome. I am personally incredibly excited that we will actually now be launching the trials of our T-cell vaccine in South Africa, where we're going to take on this mutation and go into the eye of this storm and hurricane and take on this mutation. So with that, I wanted to thank you for listening to this third series of the science behind coronavirus mutations, and look forward to presenting to you more information as we proceed further in our series on the second opinion. Thank you for being with us.